Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. This is the last lecture, and this lecture I have chosen just to discuss the summary and conclusion that we have in this Industrial Biotechnology course. Now, let us start that how we frame this course. That if you look at that, I told you that this any industrial biotechnology course has three parts. One is the bioreactor. Bioreactor is the central part where the biochemical reaction take place, we get our desired product. Now, before running this bioreactor, we required some kind of prerequisite, that is the upstream processing. Upstream processing means that you have to first you have to have the industrial microbial strain. That is, what is the industrial microbial strain? I told that whatever microorganism we isolate from the soil, that we call wild strain. Am I right? But industrial microbial strain is something different. Why they are different? Because they have the uniform this biochemical characteristics with different generation, and because when industry run use this kind of microbial strain, the reproducibility of the product, this should not be changed actually, because if the productivity change, then our production will be hampered. So that is why there is the stringent procedure that we have through which we can develop the industrial strain. That is why industrial strain is very costly. And then that for the growth of industrial strain, we required the media. And I told you media comprises of 4 different type of component. You should have carbon source, you should have nitrogen source, you should minerals and vitamins, and everything has a different purpose. Carbon, I told you that carbon goes for the cell mass formation. It also contributes for the energy generation of the process, living process. Also carbon contributes for the product formation. That is very important. Now, as far nitrogen is concerned, it mostly contribute for the cell mass formation, and your minerals and vitamins, they are the cofactor, mostly they are the cofactor in the metabolic pathway for different enzymes. So, this is so media is preparation is taking place like this, and the media for different organisms will be different. And I told you the basic difference between the chemical and biochemical process is that chemical process, that as your product changes, your raw material changes, but in the biochemical process, it so happens that same substrate can contribute to different type of product formation. As for example, that glucose or sucrose can produce for citric acid production, for lactic acid production, for ethanol production, the different production process they are involved. Now, another important upstream processing is the air and medium sterilization. I told you that our organism, desired organism should grow in the media. So, conditions should be 100% sterile, otherwise other organism will interfere in the process, and that will hamper our biochemical processes. So, because I can give a typical example that I work with citric acid industry, that and we produce citric acid by using Aspergillus niger and from the cane molasses, and as you know, cane that the yeast can grow very well in the cane molasses, and doubling time of yeast is much less as compared to Aspergillus niger. So, if little yeast that if enter into the system, they spoil the whole fermentation process. They should be perfectly sterile. So that is the air and medium sterilization plays very important role. Then that seed culture and inoculum preparation, that also plays very important role. Why? Because when we discuss the life cycle of the cell, we have come across the different phase of growth. We have lag phase, we have log phase, we have stationary phase, we have death phase. And organisms, they are very active in the log phase, and whenever we prepared any kind of inoculum for the fermentation process, it should be either in between the mid log phase to late log phase. So just to ensure that your organisms are active. So these are the different upstream processing that we have. And in the bioreactor, when you talk about the bioreactor, it uses different pumps, valve and agitator. Different types of pumps we have. We have centrifugal pump, we have peristaltic pump, because centrifugal pump we use in our day-to-day life for pumping water from the ground to the top floor of the building, and that and we have that peristaltic pump. I told you peristaltic pump largely used in the biochemical industry, because aseptically you can transfer the media from one place to others. This is that is why this peristaltic pump is large. Then progressive cavity pump is used for transferring the high thick that fluid. As for example, MADI, MADI, that suspended solid can be transferred with the help of progressive cavity pump. So we have different types of pump. We have membrane pump and different type of pumps we have. And valve also we have so many. We have a gate valve, we have globe valve, we have in the wall valve. But in the fermentation industry, for sampling, we mostly use the ball valve, because we can open it instantaneously, we can close it instantaneously, so that we can draw the sample. But we have different type of agitator. We have impeller, we have paddle type of agitator, ribbon type of agitator, different type of agitator is used, but in the fermentation industry mostly we use the impellers. So these are the different things that we have, and plus we required lot of instrumentation. As for example, we should have pH probe, we have dissolved oxygen probe, we have agitator for controlling the speed of the agitator we required, and that we know the picometer that is used for monitoring the speed of the agitator. Then we have antifoam sensor. So different type of, we have temperature sensor. So different type of monitoring system we have in the bioreactors. Now then after that, in the reactor, what we do the reactor analysis. Reactor analysis, initially I try to share with you how we can do the reactor analysis. And first I try to give the different definition of the reactor. Reactor is the vessel in which the reaction take place, and bioreactor is the vessel in which the reaction take place in presence of the biomolecules. Then when the reaction take place in the reactor, then that the rate of reaction, the kinetics of the reaction plays very important role, that this is required for the scaling up of the process. Then we shall have to find out the stoichiometry of the process. Bioprocess is very important. The stoichiometry basically gives us the information what is the maximum yield of product formation that will take place. This is very important. We discuss that. Then we discuss the enzymatic reaction kinetics, that enzymes they are very specific with respect to substrate. Enzymes are mostly they are protein in nature. Protein with active site we consider the enzymes, and this how we use the Michaelis-Menten equation just to explain the that enzymatic reaction kinetics, and later on we discuss some kind of inhibition of the enzymes, and that is largely used for several applications. As for example, for sulfur drug, we have seen that how it is used for for inhibiting some kind of organism to grow, that contaminants to kill the organisms. So this acts as an inhibitor for different enzymes. So, this is we discuss then microbial growth, product formation, all these things are very important, and substrate degradation. This is microorganism. I told you in the biochemical process that in the chemical process I told you that as your product changes, your raw material changes, but in the biochemical process same raw materials can give the n number of products, but what we change that your microorganism we change. As for example, that Aspergillus niger, it can convert sugar to citric acid, but yeast Saccharomyces cerevisiae, it can convert the sugar to lactate sugar to ethanol. We have other organisms like that different other organisms also we use for different purpose. And so microbial growth, kinetics, product formation, substrate degradation, that is we discuss in details in this particular course. Then we have given you the overview of the fermenter, how the fermenter looks, what are the different parts we have in the fermenter, everything has been discussed in this particular course. After that, we discussed that downstream processing. Downstream processing basically it is called as the purification process. When your fermenter, your bioreactor, you produce the product, that product you cannot market as such in the market. You have to purify that product. So you have to do the first you have to do the biomass separation. Then you have to different other downstream processing like distillation, that is used in case of ethanol fermentation process, crystallization, that is used in case of citric acid fermentation process, drying, this is the if your crystals are there you have to dry, liquid extraction, that is used in the penicillin fermentation process. We have different adsorption processes are there. So all this downstream processing we try to discuss in this particular course. And finally, we discuss the effluent treatment process. I told you every chemical and biochemical industry, they cannot discharge their effluent as such in the water courses, they have to treat. What are the different treatment processes and is available, and I mentioned that 70% more than 70% wastewater treatment process are controlled by the biological means. Now, after that, let me give you that what are the course content that we have in this particular course. Initially, first lecture I covered the introduction, second lecture I covered the development of industrial strain, third lecture I covered the medium characterization and biochemical pathway, which is very important. Then fourth lecture we discussed the chemical reaction kinetics. Then with this we discussed in 2 lectures, and then reactor analysis. Reactor analysis is very much required for the designing is reactor. Designing means for getting a desired amount of product, what will be the size of the reactor that you have to determine. Then we try to do the reactor analysis. What are the different reactors is there? We told you mostly we have 2 type of reactors. One is batch and the continuous process. Continuous process we have CSTR, continuous start tank reactor, and the plug flow reactor that we try to explain here. Then stoichiometry of bioprocess I told you is very important. The reason is that the stoichiometry give you the theoretical yield of the process. So that plays very important role as far as fermentation process is concerned. Then enzymatic reaction kinetics, that is reaction kinetics we discussed in 2 lectures. Then immobilization technique, I told you that when enzymes remain in suspension in a reaction mixture, that after the reaction is over, the enzymes remains the impurities that is to be taken out from the product. So what we do for the industry, they use the immobilization technique. So the enzyme contamination will not take place. Your product will be more or less free from the enzymes, and not only that, the stability of the enzymes that increases to a great extent. Then after covering all these things, I switch over to the microbial system. Initially, I discussed that life cycle of the microbial cells, which is very important. I discussed we have lag phase, log phase, stationary phase, and death phase, and every phase has its own significance. Lag phase is called acclimatization phase, log phase is called the active phase. stationary phase is called the starvation phase. So, every phase has its own significance. So, inoculum, usually I mentioned that it is usually prepared in between mid log phase to late log phase, and mostly the secondary metabolite formation take place during the starvation phase, that is the stationary phase. So, then I discuss all this thing in microbial growth kinetics in 3 different lectures. Then I discussed the overview of the fermenter in 1 lecture. Then another when you come to the industrial part of the biochemical process, the flow diagram is very important, and flow diagram may be of 2 types. One is block flow diagram and the process flow diagram. Block flow diagram means that I told you that processes are different units. There's it doesn't have only the fermenter. I have shown it has upstream processing, it has downstream processing. So, different processes are involved. How they run one after another, that is the flow diagram. And when we express the unit, one unit as a box, then we call it the block flow diagram. But when you give the sketch of the process, as the fermenter has a sketch, heat transfer equipment has a sketch. So, there are different type of like crystallizer, they have another sketch. So, if you put those sketches, then it is a process flow diagram. So, there you do not have to mention what is the process, but because that symbol tells you what is that process. So, this is flow diagram, and what are the pumps and valve used in the fermenter, that has been discussed. Then in the upstream processing, air sterilization and medium sterilization, how it is carried out, that was also discussed in details. Then I switch over to the this downstream processing, the solid separation process, evaporation, crystallizer, liquid extraction, distillation, chromatography. All this thing I try to discuss, so that that your idea about different downstream processes will be clear. After covering all these things, then I switch over to the biochemical production process, different fermentation process. First, I taking into account the ethanol fermentation process, because it started long before it was the first that so ethanol is largely used. As far India is concerned, we have more than 200 distillery industries that we have, and ethanol has a great demand not only as a beverage, but in the feedstock for the chemical industry. And nowadays the government, they are thinking seriously as a substitute of that gasoline, because the government recommended 20% gasoline can be substituted by the ethanol. We call it power alcohol. Now, after this ethanol fermentation, I discuss in 2 lectures. After that, I switch over to the brewing industry. Brewing industry that produces beer. 2 types of beer has been marketed. One is the alley beer and lager beer. Mostly in India, we have lager beer. Lager beer is it is less bitter as compared to alley beer. Alley beer we require more hops, that so it is more bitter. So, that discussion process we discuss. Then we discuss the wine industry. Wine industry is very important industry, largely that is operated throughout the world. Then vinegar production. Vinegar is nothing but acetic acid. It is a table condiment that is used in the day-to-day life. Then I discuss the citric acid fermentation in details. The reason is that that I work with the citric acid industry. I try to share my experiences that I have with the citric acid industry, and I work with a sister concern of Bombay Dyeing Group, the Citogia Biochemicals, that was located in Surat, and I discuss in details. And 2 types of citric acid has been marketed. One is called citric acid monohydrate and citric acid anhydride. After that, I discuss the lactic acid fermentation process. Lactic acid is a tremendous potentiality in the market, because I told you not only the lactic acid acts as a preservative, but also the polylactic acid has tremendous potentiality in the medicinal sector. Then glutamic acid, the kind of amino acid production I try to discuss, which is largely used in the industry. We have penicillin production. Penicillin is the first antibiotics that has been marketed, and during Second World War our soldiers are wounded and their wounds has been cured by the using this penicillin. This penicillin plays very important role in our day-to-day life. Then streptomycin is penicillin is considered as a narrow spectrum antibiotics. Streptomycin is considered as a broad spectrum antibiotics. Then Baker's yeast fermentation process I try to discuss, which is largely used in India for the production of different type of bread, and the labeling of bread taking place with the help of Baker's yeast, that not only used for labeling purpose, but nutritional quality of the bread also increased to a great extent. And then after that I also discussed the fodder yeast. The fodder is kind of that is used, Baker's is used for human consumption, but fodder is usually used for the animal feed. This is usually produced from the waste material, but mainly the waste sulfite liquor of the pulp and paper industries. Then spirulina, this is a very important algae that is largely marketed as a source of nitrogen, not only as a protein, but also it cured lot of diseases like GERDs, that the different type of problems that we have, that we can be rectified by this. Alpha amylase production has lot of industrial applications because it converts the starch material to glucose, and glucose can be used in different fermentation process. High fructose corn syrup production, this is important biochemical products largely used in the western country, particularly in the confectionery industry, and this I showed you how it can be produced. Metal leaching, that is another important biochemical application, because particularly when ore contains less amount of that metal, then we don't go for the chemical process, we go for the biochemical process, the metal leaching process that we can recover. That is already applied in the industrial sector. Then cheese making process, this is a very important application as the biochemical industries are concerned, because cheese is a kind of product through which we can preserve the milk, protein and fat for a longer period of time, because we know that milk we cannot preserve as such for a longer period of time. Hardly it can be preserved, even pasteurized milk can be preserved for 3 days, because milk is a very good media for the growth of pathogenic organism. So, but milk, protein and fat is very good for our health. So, due to the invention of this cheese, it is possible to preserve the milk, protein and fat for longer period of time. And how we preserve, because during the cheese making process one important step is the curd making, and through this curd formation process the lactic acid formation takes place, and this lactic acid acts as a preservative. That is how we can preserve the cheese for a longer period of time. Then comes to the bioenergy process, that biodiesel, that and the butanol, this is important things that is coming in the market, how we can substitute the gasoline, or biodiesel can be substitute the diesel, and biodiesel jatropha, that is largely used that for the production of biodiesel. Nowadays algae, microalgae also contains high amount of lipid that can be easily converted to biodiesel, and we found suitable for the biodiesel production. Then butanol also produced through the fermentation process. This is the old process, the acetone butanol fermentation process we know very well. That is that was also invented during the Second World War, and this is largely available in the market. Now after that I discussed the biopesticides. I told you that chemical pesticides pose lot of the pollution problem, because the whatever fresh vegetable we get from the field, that it looks very fresh, but it may contain some kind of chemical pesticide that has lot of injurious effect, because that is the kind of carcinogenic compound. So, this harmful effect can be removed if we use the biopesticides. Biopesticides, this is biodegradable, that so it will not accumulate in a system. So, it is very much acceptable to our system. Then biopolymer, in the day to day life we have seen that plastics pose lot of environmental pollution problem. It is very difficult to degrade. So, biopolymer is slowly capturing the market. We have different type of biopolymer, PHA, PLA, that two important polymer that is coming in the market, and then we just we try to find out how it is cost effective. Then I try to discuss another thing, hepatitis B. Hepatitis B kind of vaccine, this fights against the hepatitis virus, which has severe effect for liver infection. That is quite detrimental for our health. And then we discuss the insulin. Insulin is kind of hormones that is that maintain our sugar level. That is very much important for the diabetes person, is very largely used. Then I discuss the biofertilizer, that because we live in a vastly populated country and we require lot of food, and this enhancement of food production is mainly taking place with the help of fertilizer, and mostly we use the chemical fertilizer. Now, if we use the chemical fertilizer to a great extent, the fertility of the soil will be lost. I told you the fertility of the soil depends on the one of the important characteristics, the water retention property of the soil, because that nutrient take by the plant through the root, through the process of diffusion. If there is no water, they cannot take the food. So, that is the whole problem. So, water retention property of the soil is very important. By using the biofertilizer, it is possible to increase the fertility of the soil. So, this is very important. This is slowly capturing the market. Now, I told you that as far effluent treatment process is concerned, that biological effluent treatment process playing very important role. 70% of the wastewater treatment process are controlled by the biological means. One is this aerobic effluent treatment process, and then I also discussed the anaerobic fermentation process. Now, here I want to stress one thing. Initially, we considered the wastewater treatment process is not cost effective, because every industry they consider 10% of the total investment for the wastewater treatment process, and they don't consider any kind of return from that. But due to the advancement of the anaerobic digestion process, now it is possible to get back some money from that. Now, how we get back the money? In the form of the methane and digested material we can use as a good biofertilizer that can be used for the in agricultural field. So, this is how we can add some kind of value to these processes. That then we discuss about the 10 cubic meter pilot plant operations for the biohydrogen production, that we already discussed in details. We have given videos here, that so I hope the main purpose of discussing this is that just to give you the impression that how a process in industrial level can be operated. This in IIT Kharagpur we have fabricated, we have designed and fabricated and erection of this whole plant, and in this particular in the institute, and we try to share with you how we operated. So, this is when you go to the industrial sector, it is more or less same, only it is little bit in a bigger type. So, this is the thing that we try to discuss in details, and this lecture we try to share with you the summary and conclusion. So, so these are the different reference book that you can consider, that industrial microbiology by Prescott Dunn, this is very important book. Then biochemical engineering fundamentals by Bailey Hollis, then bioprocess engineering principles by Duran, then bioprocess engineering basic principles Suller and Gargi, biochemical engineering by Iva, Humphrey and Millis, a textbook of industrial microbiology by Kruger and Kruger, the chemical reaction engineering by Liebenspel, and I have written 3 books that also can be consulted. One is Biohydrogen Production Fundamentals and Technology Advances, Biohydrogen Fuel for the Future, and the Algal Biorefinery and Integrated Approach. This is the books that we have published. This is published by CRC Press, and this book, the biohydrogen is nothing but hydrogen followed by methane. This has also, we find this is the process will be with us for a longer period of time. This also we have published, and this is the first book that has been published in the world. And then we also publish another book, that is the Algal Biorefinery. We consider algae can be used for different purpose, not only as a source of it is considered the 3rd generation biofuel, but it not only considered as a fuel, but also it can be considered as a source of protein. It can be considered as a as a source of pigments. It can be used as a for the source of biochemicals. Now, in this particular lectures, I have this is the total number of assignment is 12, and 1 assignment per 5 lectures. This is common to all NPTEL courses. Now, here I want to point out that this is the main outcome of the course. The first we try to you should have some kind of information or block flow diagram and process flow diagram of industrial fermentation process. Then to understand the different unit operation involved in the fermentation processes. Then development of the expertise on the analysis of reactor used in the industry. Development of the seed culture in the laboratory, development of inoculum for the industrial for the production fermenter, the product purification, quality control of the fermentation industry, material analysis of the fermentation process, biological wastewater treatment process, use of valve and agitator. These are the different outcomes that information we try to keep in this particular course. So, I have 2 teaching assistant in my course. One is V. Ramaiah, that her background is BTech from Biotechnology from IIT Durgapur, currently pursuing the PhD with me, and her email ID is given here. Another is Chandan Mahato. He did BTech in Chemical Engineering from IIT Durgapur, currently pursuing MS at IIT Kharagpur under my guidance, and his email ID is given here. So, I am from IIT Kharagpur. I am here for last 31 years, and I hope that you have enriched your knowledge in the area of industrial biotechnology, and I try to share my industrial experience with you, since I have been teaching for 31 years and I work with the industry for about 2 years. So, how I try to make a bridge between the academic and the industry. I think this will help you in your future career. I wish you all the best. Thank you.